Okay, good to see you all, and thanks very much to Jing and also Dongwei for hosting me here. So this is my second time visiting Zhuhai, and I really enjoy it, and also the beautiful campus and the surroundings, but also the discussions with people here. And it has also been a great pleasure to have the course over the last 4 years and meeting the students from here, Guangzhou, and also from Beijing. Also, thank you to the students for the lychee fruit. I'm very much enjoying them. So today I'm going to talk about our latest work on visual exploration and reading. I have some new data to show that I have not been presenting before, so I'm very much looking forward to also hear what you have to say about it. Nevertheless, so a more recent focus in our group has been visual exploration, meaning vision with saccades. And the idea is that what we want to study is not only human vision, but also human vision when people are doing active saccades. And that's also to get more close to having so-called ecologically valid paradigms, meaning paradigms that's sort of more too close to what we do in real life, right? And often over the years, when we are doing psychophysical experiments and vision experiments, we ask people to fixate, and then there might be a cue, and a few seconds later, there's a sort of attention allocated to the left and the right. But these tasks are very artificial, right? And in particular, we ask people not to saccade. But as it turns out, we are typically saccading 3 to 4 times per second. So the claim here is by asking people not to saccade, we are missing something very fundamental about vision. When we think about vision, it has been known for many decades that typically people move the eyes to important parts of a visual image, right? So now it turns out that there's a bit of a conundrum here, because how come we can move our eyes to important parts, so to speak, of a visual scene already before we know what that object is, right? So how does the visual system solve this? Well, part of the solution is that objects out here, before we saccade to them, have to be pre-processed, right? So, of course, these objects, when we fixate here, these objects would be, for instance, in the parafoveal. So then we can talk about parafoveal processing. And the question is, how much information can be derived from the parafoveal already before the participants are moving their eyes? And so what you see here is to add to the mystery, in a sense that we know that we saccade 3 to 4 times per second. That means we have 3 to 4 saccades within a second. So that also implies that a lot has to be processed very fast, right? So if we are saccading around here, for instance, if you fixate on the woman and then is about to prepare a saccade to the dog, then not only do you have to process the woman, but you also have to prepare your saccade to the dog. And all this has to explore potential saccade goals. And all this has to happen within 250 milliseconds. Since we know that it takes about 100 milliseconds to sort of prepare and initiate saccade, there leaves only 150 milliseconds to both process the object you fixate at, but also entertain future saccade goals, right? So there's a need to really study on a fast time scale what is going on in the brain when we are doing this natural visual processing. And of course, to do that, we need some tools that allows us to investigate this fast processing. And in our case, that's MEG, right? So one can investigate this in humans with EEG and MEG. So the first study I will show you is on natural visual processing, exploring these kind of objects. So this is a study done by Camille Fakche. And the task here is that people can move their eyes from object to object as they like. Then 2 seconds later, there's a set of objects again, and then they have to identify the object that was different from before, in this case, the cat, right? So this is just a task enforcing that people are actually sort of trying to remember the objects and move the eyes amongst the different objects. So these objects are different categories. For instance, it can be animals and food items, fruits here, right? Furthermore, what we also did was to have some of these objects in color or some of these objects in black and white, right? So that means we can now look at feature processing, meaning the color, and we can also consider object or category processing, namely sort of, you can think about that as semantic processing and sort of processing the meaning level of these objects. So with MEG, we can study this, and you've probably all heard about classification approaches using support vector machines, right? So the idea is that we collected data, in this case from 306 sensors at the same time, and then by the brain activity, we ask, in this case, whether we can predict whether people were looking at a color object or black and white object, right? And this is an example where we just consider 2 sensors, and based on these 2 sensors, we might be able to tell whether a color or a black and white object was perceived. But of course, the more sensors we have, the better we can do. So this is just 2 sensors for illustration, but of course, in real life, we have 306 sensors. We can do the same with category. So in this case, we can try to tell fruits from animals. And again, using our 306 sensors and a support vector machine, we can then predict from the data whether people were looking at fruits versus animals. And we like to think that if that's possible, then we can derive the brain activity associated with semantic processing. So we have the tools for that, and there's been a lot of prior work showing that this is indeed possible. So what we now have been doing is that when people are saccading around between the different pictures, what we are considering both the foveal, meaning the fixations, the fixations, and then the objects associated with the fixations. Can we decode that, right? What we're also considering is upcoming parafoveal images. So that is the next saccade goal. So when we fixate here, how much can we predict of upcoming objects? However, we are also considering past parafoveal images. So could it be that there's some sort of maintenance of what you just looked at before when your eyes are moving on to the next object? All this we have been trying to quantify. First, a few words on the saccades. A saccade duration when doing this task is 250 milliseconds, meaning about 4 saccades per second. And the duration of the saccade itself is about 40 milliseconds. So, and we have about 10 to 15 fixations per trial, right? So we have plenty of data to analyze. So here comes the first result. This is when decoding what people actually are fixating at, right? And then you already know from the literature that is possible. And what you should appreciate here is at about 150 milliseconds, 145 milliseconds, there's a peak in the decoding of the category of the objects, also consistent with the literature. However, when we consider the decoding of the feature, meaning the color, we can do that at 70 milliseconds, right? So it seems that there's sort of a sequence of processing going on in the brain. First, the feature level is being decoded, in this case, color. It might be before. And then there is the category being decoded, might be sort of downstream IT, maybe in that anterior temporal lobe at 145 milliseconds. Okay, so this we already know would be possible. What we don't know is to what extent is it possible to decode what is in the saccade goal in the parafoveal. And this is what you see here. So of course the decoding level is lower, but it is indeed possible to decode both the color and the category of these upcoming objects. Yeah. Yes, that's correct. So what you see here is that since we are actually saccading around, and before we did the fixation, our eyes were actually moving to that object. Turns out that we can actually decode the category already before our eyes land on this, and then it peaks later. So this is already evidence for some sort of parafoveal processing. So we can also make that case, as I will do now, by seeing when can we decode the upcoming parafoveal objects, right? And it turns out we can do this. So this decoding result is consistent also with these pre-saccadic decoding results.
Here the color is more clear in parafoveal.
Yes.
But foveal cases, you can see the color is not significant.
Yeah, that's correct. So the way we interpret this is that indeed color can be decoded in the parafoveal, but it's not particularly sustained. And then a little later, so you decode color in the parafoveal at 88 milliseconds, and then you decode category at 160 milliseconds. But what the brain really cares about is the category of the object, and color is less relevant. So then we like to think that the information about the color sort of fades out, and that's why you see the category decoding out here. Yeah. Okay. So we'll discuss this timing in a minute, but I should also say after our eyes move away from an object, meaning then we talk about the path parafoveal decoding. There's also some sustained multivariate activity reflecting that, right? What this all means is that we have foveal decoding, upcoming parafoveal decoding, and past parafoveal decoding. And this probably reflects that when you see a full scene, these different objects sort of have to be typically have to be sort of maybe not integrated, but sort of you need to form a percept of the different objects also in regard to their different locations. Okay, so let's now discuss the time course of this. And this is because one might ask what's going on in terms of sequential processing and parallel processing, right? So this here actually suggests that multiple objects are being processed at the same time, what you fixate at, but also future saccade goals. So let's think about how the brain does this. Could that be sort of a sequential mechanism where you first process the poppy, and after that the fruits out here, right? Or could it be that you process this in parallel, namely at the same time? Well, the answer is something in between, in the sense that if you look at the color decoding in the fovea and then the parafoveal, you see it at 70 and 80 and then 88 milliseconds. So that sort of speaks to it being sequential. If you look at the decoding of the category, that is 145 and then 160 milliseconds. So that's also somehow sequential, but there's also overlap, right? So what we would like to think is that there's sort of a mix between sequential and parallel processing. And I'll try to make sense out of that in a minute. So this is just to show you statistics of these numbers. When we look at the statistics, the category difference is significant. The color difference in timing is barely significant, right? So mainly this effect between 145 and 160 is the robust one. Nevertheless, how do we interpret these findings? So we like to think that there's some sort of pipelining mechanism going on in which different objects are processed in parallel, but at different levels of the visual hierarchy. So what do I mean by this? Well, it's in fact not so complicated if you think about doing the dishes, right? So this woman, she's washing the plate, and then she passes it on to the man who's doing the drying. So these two people are working in parallel, but they work at different levels of the processing hierarchy, right? So it could be, if we make the analogy to the processing of objects, there is feature or color processing going on, and then later on category processing, right? So I'll get to, so basically this is the notion we think, and we have an opinion paper out in Trends in Cognitive Neurosciences where we have made these mechanisms explicit. So if you go back to the picture here, if you think about processing the woman and then the dog, what we have proposed is this pipelining mechanism where there's feature processing and category processing, meaning color and category, semantic processing. This feature processing is sequential, and the category processing is sequential, but nevertheless, there's pipelining going on. So while the category of the woman is being processed, the feet, meaning the object you fixate at, the features of the dog that you're processing is going on at the same time, right? So this is what we mean by pipelining. So this is early days, so we don't have fully evidence for that, but at least the data are consistent with this. We don't have strict serial processing. We do not have strict parallel processing, but possibly we have pipelining going on. So to conclude this first part of the study, what we can show is that we can process both feature and category in the parafoveal of objects before we saccade to those, right? So there has already been some indications in the literature demonstrating that's possible, but now we also have the time course for this. And we can show that this goes on relatively fast. We have this sort of early support for a pipelining mechanism in which different objects are processed at the same time, but at different levels of the visual hierarchy. Okay, so so far so good. So the next topic I want to move on to is reading. So before we talked about exploration of visual objects, however, one could consider reading as a special case of this visual exploration, right? Of course, reading is a very important task. In order to achieve academically, then we need to have good reading skills. Furthermore, reading is a skill that we are not born with, right? It is a skill that is unique to humans, and it's a skill that we have acquired through developing languages and then later on written languages, right? So it's a very special skill unique to humans. And furthermore, this is a task that the children learn at the age of 4 to 8, right? So different countries start to teach children to read at different ages. But of course, it's very important to understand more about this reading or these mechanisms supporting reading in children. In a sense that some children also have problems learning to acquire reading skills. We can also think about then what can be done to help these kids. But in order to do so, we need to know more about the supporting mechanisms. What I will show you now is some studies we have conducted in adults, but later on we want to take that to children. And the ambitions of this project might also align quite well with the China Brain Project, where now I understand from Jensen that data are being recorded from thousands of children. Nevertheless, when we read, just as when we are doing visual exploration, our saccades, we make saccades about 250 every 250 milliseconds, so about 3 to 4 hertz, right? Furthermore, there's also this parafoveal processing going on, as we talked about before. What is the evidence for that? Well, the evidence is that sometimes we are skipping words, right? So the quick fox jumped over the lazy dog. Sometimes you will find that when people read sentences like this, then typical words like the is being skipped because it's not very informative. But in order to know that a word in the paraphobia is not very informative, it needs to be processed, right? So basically what we are going to do is that we're going to translate what we know about saccading during visual exploration to reading. And in fact, reading is somehow a more simple case in the sense that the saccades are in one dimension. And furthermore, often the stimuli, meaning the words, are well characterized. So the first study I'll show you is done by Yali Pan. She did her PhD at the Chinese Academy of Science, and she has been a postdoc in my group for quite some years now. So one of her studies has been on the semantic previewing when people are reading, right? So in this case, the quick brown fox jump over the lazy dog. When we then read the word fox, one can talk about semantic processing. And semantic processing is referring to deriving the meaning of fox. So we know when we think about a fox, that is an animal running around in the forest. We know about the color, they're sort of reddish brown and so forth, right? So there's all these features associated with fox. And when we read the sentence, we need to derive those semantic features and then integrate that into the sentence context, right? And again, this has to go on a very fast time scale, since we only have 250 milliseconds per fixation. Again, 100 milliseconds are devoted to the saccade. So within 150 milliseconds, we have to derive the meaning of the word, integrate it into the sentence, but we also need to prepare the future saccades, right? So how is that done? Well, to investigate this, we have been making use of a new technique we have developed in my group. It's called rapid invisible frequency tagging. And this is based on this projector. So it has a very high refresh rate, meaning we can flicker individual objects in a scene or words at very high frequencies. And the idea is, as you see here, we have the projector, we flicker the stimuli at 60 Hertz in this case, and then we can measure the luminance of that flicker, but we can also measure the brain response. So now it turns out that it's possible for the visual cortex to follow these flickers at 60 Hertz, in the sense that we do get a sizable signal. What's also good about this is that you do actually not perceive this flicker. So this is above the so-called flicker fusion rate. So people are blind to this. So we can manipulate that visual scene with this flicker without people being able to detect it. And we have done previous studies where we show that spatial attention can modulate this flicker, right? So if you attend to something out there in the paraphobia, then it's associated with an increase in the neuronal excitability, and that is indicated by an increase in this flicker signal. So we have been doing this in a reading study where the eyes are jumping from word to word. So this is just to show you how well it worked. This is a measure of the response. We get a strong response in the 60 Hertz frequency tagging over visual cortex. We can localize that to early visual cortex. This graph here shows the flicker response from sentences, and it is word N that we flicker, right? So when you fixate at that word, you get a fairly strong response. The interesting thing is that if you fixate before that word, you get an even stronger response. If you fixate after that flickered word, you get a smaller response. So there's this asymmetry reflecting that we are moving our attention to the right all the time when we are reading, right? Probably not a big surprise, but here we have sort of a neuronal signal reflecting that movement of spatial attention towards upcoming words, right? You can also think about this as covert attention, even though there's a debate of whether there's a difference between covert attention and pre-saccadic attention. Nevertheless, think about this as being attention being moved to the right, and we can index that in terms of neuronal excitability as measured by the frequency tagging signal. So how do we study the semantic component of this? So what we do is that we have sentences in which there's a word being incongruent with the prior part of the sentence. So Lily says this blue brother will be a big fashion trend, right? This doesn't make sense. Brother, so blue brother doesn't make sense. Lily says this blue jacket will be a big fashion trend. That makes sense, right? So the question is, when we have words that do not fit into the sentence context, how does that reflect the frequency tagging? And of course, in order to know that this word does not fit into the sentence, you need to process that word at the semantic level, right? So the big question here being that when you fixate on blue and is about to saccade to brother, how does the frequency tagging signal reflects the congruency? And of course, it's the target word here, brother or jacket, that we are flickering, right? So we are really interested in this flickering signal when we are fixating on the pre-target, right? And you see the results here. So this is a bit of a complicated figure, but the first time frequency representation here reflects people fixating on blue, and then the red blob here is the response from brother being flickered. What you see below is the same, but this is when jacket is being flickered. So now we can compare the frequency tagging response for the incongruent versus the congruent sentences, right? And the funny thing is that there is a reduced flicker response when the word is incongruent with the sentence. So we'll discuss what that means in a second. But nevertheless, this provides evidence for there being attention allocated to the words in the parafoveal. And this attention allows you to process the word in the parafoveal at the semantic level. And that is evidenced by the difference in the frequency tagging signal. And very importantly, we see this signal already 100 milliseconds after fixation on the pre-target word, right? So it happens very fast. It happens so fast that we actually argue that this processing of brother or jacket happens not only when you fixate a blue, but probably when you fixate on the words even before that, right? The interesting thing here is that there is a decrease in the frequency tagging signal for the incongruent words. So it's almost as if your attention is being repelled by the incongruent words. It's almost like your attention is sort of being so it's almost like when you discover you cannot fit that word into the sentence context, you don't attend to that further. So then you might ask, does this have consequences for how well you read? So what we now can do is to relate this difference in frequency tagging to the reading speed, right? And what you see here is a figure where we have the difference in frequency tagging on the x-axis and the reading speed on the y-axis. So a participant out here is a relatively fast reader, but that's also a person who has the stronger parafoveal effect at the semantic level, right? So this suggests that people who do more of this semantic parafoveal processing are also more efficient readers, right? So this is, of course, also going to be important when we are linking to reading abilities in children. So I'm going to conclude the first part of this study, where we have been using this new technique of rapid frequency tagging. So frequency tagging is not new, but using frequency tagging above 50 Hz is. This has now provided evidence for semantic parafoveal processing, and furthermore, this processing is reflecting reading speed. These results are about to come out in eLife. We also have a previous study on this in Nature Communication, where we look into lexical effects in parafoveal processing. So I'm going to switch to another study where we have complementary results. So this is done by Lichuan Wang. So she used to be a master's student at Guangzhou. Yes. Yes.
Task is the nature reading task.
Yeah.
And different subjects they set at two different places. Does the distance between the previous word and the pattern word matter? Not all the subjects have to do all this. Someone may have to do this, and someone may have to do.
Yes. So it's a good point. So what we have done is to have a lot of sentences, and then we some subjects would indeed skip a common word like this. So I don't remember the details, so we have to ask Yali Pan, but I think we took that into account as well. But what we really, we of course rejected trials where people did not fixate on blue. It should also be mentioned, sometimes people do sort of multiple saccades on the same word, sort of adjustments of saccades, if you will. So we only took the first saccade into account. So there's a lot of details to the trials we kept and not kept. Okay. So the next study is done by Lichuan Wang, who used to work at South China Normal University in Guangzhou as a master's student. Now she's a PhD student funded by the China Scholarship Council. And what we have been looking into here is both orthographic and semantic processing. And now we are using multiple analysis using representational similarity analysis. And the idea here is that, again, we have natural sentences, and then we also do MEG and eye tracking as before. So here we have been doing a trick in the sentences we have been constructing, in the sense that we are making sentence pairs with different properties. So some of the properties are overlap in orthography, right? So police and policy, of course, mean very different things, but they have a big overlap in terms of letters, right? So they only differ by the last letter, right? So what we now can do is to consider the brain activity associated with the similarity for these word pairs. So the idea is that by simply looking at the sensor activity and correlating that, we can ask if the sensor activity is more similar for policy and police versus policy and writer, which looks very different, right? We can also do the same in terms of semantics. So police and guards have very sort of similar semantic meanings. So that could be people in uniform exercising control, right? So now we can look at the similarity, meaning how well the brain activity correlate for guards and police. And then we can compare that to between pair, police, and author, right? So this is all there is to it. And we do that with respect to when people, they fixate on the word clever, right? Because when you fixate on clever, then we can ask how much is done in terms of parafoveal processing at the orthographic and the semantic level. And this is, again, how we do the analysis. As I mentioned, we do a representational similarity analysis. And basically, we are simply looking into the brain activity. So these color squares, they reflect 1 to 306 sensors. And then we simply do the correlation brain activity for these respective word pairs, right? So while representation similarity analysis might sound complicated, it's basically nothing more than doing simple correlations. And then we can track these correlations as a function of time and compare them for within pair and between pair features. And this is the first result in terms of orthographic processing. And what you should appreciate here is about 100 millisecond in, when fixating on the word clever, we see a fingerprint of orthographic processing because correlations are higher for within pairs than between pairs, sort of within orthographic pairs like police and policy, right? And this difference here is robust, and we see it, as I mentioned, about 90 to 100 milliseconds in. Yeah, right. So this is evidence for words being processed in the parafoveal at the orthographic level, right? So this might not be a big surprise, but the news here is that we can derive time course of this. What's also new is that this parafoveal processing at the orthographic level is predicting reading speed. So this person out here is a fast reader, but it's also a reader with big difference in the correlation, meaning more parafoveal orthographic processing. Since we are doing MEG, we can also localize that effect, and we see a strong source of this in the left visual word form area, right? So this is really a nice confirmation that this works quite well. So we talked about a lot about methods the last 4 days, but the way we did this analysis was to take the data to source level. So we make we extract the surface of the brain, we do a grid, we do some spatial filtering to derive the time courses at each grid point, and then we redo the RSA analysis at source level using a searchlight approach, and then we find the effect in the visual word form area. There's also an effect in right temporal cortex we don't really know how to interpret. Okay, so the next question is, what about semantics? This would be looking at the brain activity, or considering the brain activity for, say, police versus guards versus, and then comparing that to a between pair set of words. And again, this is derived when people look at the word clever, the pre-target word. And here we see a difference at about 130 milliseconds. Again, robust. And this difference reflects the semantic parafoveal processing, right? So now we have convergent evidence with the rapid frequency tagging and the RSA analysis that parafoveal processing can be done at the semantic level very fast. Here it occurs at about 130 milliseconds. Furthermore, this semantic parafoveal processing is predictive of reading speed. So this participant out here has a big difference in the correlations, but is also a fast reader. Okay, so again, we can localize that activity, and this activity is now localized to the left inferior frontal gyrus, right? So this is again done by a searchlight approach applied at source level. So the left inferior frontal gyrus has, of course, been implicated in language processing, but also in semantic processing. So I'm going to finish this part by concluding that we now have convergent evidence for semantic processing. Furthermore, using the RSA approach, we can localize the semantic processing, semantic parafoveal processing to left inferior frontal gyrus. And we also find evidence for orthographic processing in the visual word form area. And both these effects are predictive of reading speeds. Okay, so far so good. Then the next topic is going to be on the timing of it all. And again, this is back to pipelining, right? So I mentioned pipelining before during natural visual processing. We also think that pipelining is going on during natural reading. And again, this is about operating at different levels of the processing hierarchy, but doing these operations in parallel. So in terms of word processing, what we think is going on is that the word jumped, you fixate on that. So word jumped is being processed for orthography first in visual word form area, and slightly later for semantics, right? So I should actually move this line to left inferior frontal gyrus. Anyhow, so the word jumped is passing through the processing hierarchy. But as soon as jumped has been processed for orthography, that frees up resources for the next word, namely over, to be processed, right? So this is the parafoveal processing at the orthographic level. And then that moves down the hierarchy. And then eventually that allows for the word over to be processed at the semantic level, right? So again, for reading, we also think this pipelining mechanism is going on, in which different words are being processed at the same time, but at different levels of the processing hierarchy. So furthermore, when you think about this pipelining, this acquires timing, right? So of course, if the woman passes on the plate to the man before he strikes the plate, there would be trouble, right? So these two people have to sort of coordinate their processing, if you will, quite tightly. So how is that done? Well, we would like to think that brain oscillations are important for this, in particular alpha oscillations. And this is quite a different way of thinking about alpha oscillations if you're used to think about alpha reflecting inhibition or rest, right? But we can discuss later maybe while this notion here is not inconsistent with the inhibition process of alpha oscillations. In fact, we think that these alpha oscillations is coordinating by means of pulse inhibition. But the key point being that if we think that these alpha oscillations are clogging the processing and coordinating the pipelining, the saccades themselves also have to be locked to the phase of the alpha oscillations, right? So what we would like to think is that our saccades are locked to the phase of the alpha oscillations such that the visual information is hitting the excitatory phase of the alpha cycle. And this is also motivated by the fact that we saccade 3 to 4 times per second. But if you think about the alpha oscillations being a clocking mechanism of visual processing, these timescales actually fit together in a quite awkward sense, right? So if you have to do these saccades 3 to 4 hertz, it seems to be silly to design a visual processing system that has a clock frequency of 10 hertz, unless these saccades are locked to the phase of the ongoing alpha oscillations, then things can start to make sense again, right? So I'll now show you some support for this actually being the case. Before that, I'll just show you a study that we're quite excited about, done by ECOG recordings in the marmoset monkey. Basically, what you see here is a traveling wave. This wave is elicited by saccades. This wave is at 8 Hertz, so in the alpha band. So each time the monkey makes a saccade, you see this swoop of activation going down the ventral stream, right? And this is exactly the swoop of activation we think that is organizing the pipelining in humans as well. However, this requires that we then also can see saccades being locked to the phase of ongoing alpha oscillations. So the first study I will show you here is a study of people doing visual exploration. So then, of course, we can quantify the saccade as a function of time and then relate it to the ongoing brain activity. So this is a task where people have to study natural images and then later they are probed, right? So not to go too much into the details of the task, but the point is that we can consider items that are later remembered versus later forgotten. And what we then have been looking at is the ongoing brain activity, and then we have been relating the alpha oscillations to saccade onsets. So we can consider one saccade, another saccade, and a third saccade, right? So basically, we have all these saccades, and then we look at the ongoing brain oscillations before the saccade to ask, is the saccades locked to the phase of these ongoing oscillations? The way to test for this is to derive the phase of the alpha oscillations prior to the saccade, and then ask, are these phases aligned, right? So we have been using some kind of phase locking index to do that. And when doing this, what we find is that indeed there's around 12 Hertz oscillations being locked prior to the saccade or the left posterior cortex. So the best way to interpret this is that you have ongoing oscillations and then you saccade at certain phases of these ongoing alpha oscillations, right? That means that the alpha oscillations are the boss, so to speak, and they determine when a saccade can be released. Furthermore, we see a stronger phase locking effect for later remembered versus later forgotten items, right? So there's also a functional importance to these alpha oscillations. Furthermore, we can localize those to the hippocampal, parahippocampal, and retrospinal areas, right? So we think that these saccades might be involved in linking, in this case, the visual system to the hippocampal memory system. So as you say, we also reproduce these findings using intracranial recordings in human participants, right? So as you say, this is a fairly robust effect that we see both with MEG data, but we also see it by intracranial recordings. So the next question is, do we have a similar effect during natural reading, right? So this is, again, a study being done by Yali Pan. This is, in fact, our previous study that we reanalyzed. So the key pointing being that if you just look at the time frequency representation of power during reading, and we then cut out snippets of data around each saccade and do the time frequency representation per snippet and then average that up, what we see is there's relatively strong alpha power during reading, right? So this might be a bit of a surprise because we are sort of used to the fact that when you flash something on the screen, alpha is being depressed, right? So this is called alpha blocking. But in fact, when we are reading or doing visual exploration, there's not this strong onset of visual stimuli. There's just the eye moving around, right? So in that case, we do not get strong alpha blocking, right? So if you think that alpha is gone during natural visual processing and reading, then you are incorrect in the sense that we have strong evidence for alpha still being present during these natural visual processing events, right? And of course, this is important in the sense that we have been investigating alpha for now a few decades. And if these alpha oscillations are not related to visual processing, they are less important, right? But now we actually find they might be related intimately to visual processing. But again, the important part is how they link to saccades. So this is exactly the same analysis where we look at the pre-saccadic phase of the ongoing oscillations, and the findings are very clear when we compare saccades towards low versus high frequency target words. When we look at the saccade onset prior to the saccade, we see this alignment of the alpha oscillations. So again, this suggests that the alpha oscillations are in control and they allow saccades to occur, but only at certain phases of the alpha oscillations. I should also stress here that saccades are not rhythmic, right? But the saccades are locked to ongoing oscillations, but not every cycle, right? So there might be there might be 3 cycles, 2 cycles, 4 cycles between each saccades, right? But when you saccade, the oscillations provide an window opportunity for when the saccade can be released, if you will, right? So in this case, the effect localizes to right posterior regions, Brodmann area 7. So this is also a region that has been involved in saccade planning. So I'm going to conclude the alpha and saccade findings. So we have convergent evidence both from visual exploration and reading that saccades are locked to the phase of ongoing oscillations. This logging is dependent on sort of the performance or difficulty of the task, right? So if people are remembering better, there's more logging. If people are saccading to a low frequency word, meaning a difficult word, there's more logging, right? So there's also a functional component related to the task in terms of this phase logging. So the last part, what I'm going to talk about is reading in children. So we have no results on this, I should say upfront, but this is something we are working hard on. So there's a lot of interesting questions pertaining to reading in children, right? First of all, not so much work has been done using EEG or MEG on reading in children. So there's a big question on when do children learn to read fluently, in a sense that fluent reading we here consider as the ability to take in multiple words at the same time, right? So a lot of work has been done on teaching people to read one word at a time and become proficient at this. That's all well and good, but we would like to argue that real proficient reading is associated with taking in multiple words at the same time. So when does that start? We do not know. Now we have the tools for investigating this. Furthermore, there's an interesting aspect of alpha oscillations. It turns out that when children are about 4 to 5 years old, the alpha oscillations are slower in the theta range, but as they get older and become early teenagers, these alpha oscillations accelerate to the alpha, to the sort of the adult alpha band, right? So actually, it is exactly in the age range when alpha oscillations accelerate in frequency that children also learns to read. So one can, for instance, now ask, does the ability to acquire fluent reading rely on the alpha oscillations being mature in terms of frequency? That's something we are going to investigate. And of course, all this insight can be applied also to understand reading disorders and maybe find strategies to better alleviate those problems and help children that do not read so well to become better readers. Okay, so now there's one problem here, and since our core tool is MEG, a conventional MEG system is not great for young children because their heads are too small, right? So they simply bob around. So nevertheless, there's a lot of excitement in the field on these new type of OPM sensors. And I also know here in Zhuhai, you have a new OPM system. And and also in Birmingham, we are developing an OPM system that can be used for children. And we do this because we want to investigate reading. And the big advantage of these sensors is compared to conventional imaging, they do not require cooling by liquid helium, right? So that means they can be placed close to the head of these children. So we are in the process of developing such a system. We now have a system up running, but now we are taking the next step of designing the part for children. So our OPM lab is in particular spearheaded by Anya Kowalczyk. So she's a physicist, so she can also develop her own sensors. So we're also working on, or we have just submitted a paper on a gradiometer, an OPM gradiometer. But nevertheless, the system we are developing for children is relying on commercial sensors. So we're quite excited because now we hope to have the ability to take our reading studies conducted in adults to investigate reading in children. So I'm going to summarize here. So I think we now have good evidence for semantic previewing, both during visual exploration and during reading. Furthermore, we also have evidence that saccades are locked to the phase of ongoing alpha oscillations. To us, this is consistent with this framework of pipelining, where several words or objects can be processed in parallel, but at different levels of the visual hierarchy. And furthermore, we think that this pipelining is organized by alpha oscillations. We still need to do a lot more work to substantiate this theoretical framework, but that's what we are working towards. So in the future, we therefore hope to find more evidence, empirical evidence for this pipelining mechanism. We're also building a more rigorous computational model for this. We also would like to investigate how the oscillations are causally important. So we hope to do perturbation of the frontal eye field with TMS or maybe ultrasound to investigate this. In the past, we have shown that if you do TMS to the frontal eye field, you disturb people's ability to modulate the alpha oscillations. And we would like to apply all this to investigate spatial attention, in particular reading in children. So I will end here by acknowledging my research group, who are doing amazing work on all these topics. Thanks very much for your attention.
Yeah, I mean there is a saccade suppressing phenomenon in visual search, and from your study, is alpha oscillation enhancement cause the saccade suppression? I mean how I mean, from saccade studies, we find the saccade separation, which means that there is a blend and block during the saccade.
Yeah.
And you find the alpha oscillation enhancement if you observe to the saccade. Is there any relationship between them? And also from your reading studies, I think also have some kind of results during the saccade. And what kind of content is blocked during the saccade?
Yeah, no, that's all excellent questions. So as you mentioned, during saccades, there's the saccadic suppression, right? So at least what we would like to do is to entertain the possibility that since we think these alpha oscillations are consequences of pulse inhibition, then the saccadic suppression is also a consequence of the alpha pulsing in an inhibitory sense. So we have not evidence for that yet, but that's something we would like to work towards. And then the idea is that when the saccadic suppression dies out, there's this downgoing ramp of removal of inhibition that then allows the most excitable visual items, in the case the words, to activate according to excitability. So you get this sequential activation, right? So it's something we would like to investigate. But I should also mention that when we are doing the frequency tagging, we actually do not see too much evidence for the saccadic suppression. So it's a bit of a mysterious topic. And also when we do the RSA, the multivariate analysis, we don't see evidence for the saccadic suppression either. So it could be that the saccadic suppression sort of belongs to the early visual system, whereas the, say, semantic formation, or maybe even orthographic information is carried through during the saccade. But it's all good questions that we hope to investigate. Yes?
I think the alpha oscillation would only relation to the cut. Or it just also the something next to this potential shift.
Yeah, so I mean, it might be doing all these things, right? So in the past, we have been doing a lot of work showing that alpha is reflecting a location of attention. So if you attend to the right, your left hemisphere alpha goes down and your right hemisphere alpha goes up. So the issue now is that during reading, it's hard to investigate because you only saccade in one direction. So we cannot do sort of the left versus right attention saccade. But I mean, there do seem to be sort of a tendency to your left hemisphere alpha being depressed compared to your right hemisphere alpha. But it's probably not a big surprise, right? So there's these power modulations of the alpha oscillations, but I think what we have to get more serious on is investigating the phasic element of the alpha oscillations, in particular when we're operating on this fast time scale. So the reason for why we're also getting into this is that we indeed have been doing all this work where we ask people to attend to the left, then we wait a second or two, and then we put up the stimuli, and then we look at the alpha band lateralization, and then that alpha band lateralization might comes on after a few hundred milliseconds and then stay on for a good second, right? So the problem about all this work we have been doing is that if you saccade 3 to 4 times per second, maybe all that work is less relevant, right? That's also why I think it's so important to study these more ecologically valid paradigms, in this case with saccades, because it sort of really forces us to rethink what mechanisms are important or not. And I myself have been working a lot on the alpha oscillations and how they are modulated by attention, but maybe some of this work is not super relevant because it's on too slow timescales to actually impact what the brain is doing in real-life situations. That being said, this sustained alpha lateralization could be important for visual exploration and reading, in the sense that the alpha oscillations then are sustained and sort of shifting their attention to the right. But I really think it's the phase and modulation of the oscillations that we need to focus on. Yeah.
What depends on children, like children that is a right approach. Just remember, you know that about 5% people are TikTok.
Yeah.
That's it. Maybe it won't be like say adults.
Yeah.
Yeah. All those that use healthy adults.
Yeah.
That's 5%. Not very adults.
Yeah.
So if why I know that's just
Yeah. No, that's a good point. So you're suggesting to when studying dyslexia, start with adults because it should be possible to find a good group of those. Yeah. Yeah. No, that's certainly a good starting point. That's something we should consider. And so I didn't mention dyslexia, and that was partly on purpose because dyslexia is a complicated topic, right? And there's a big discussion on also the phonological component on dyslexia. Other people argue for spatial attention problems, right? But I think the sort of phonological view on dyslexia is the dominant one. Nevertheless, we hope to integrate that also by looking into parafoveal processing at the phonological level, right? So there's also debate when you're reading a word. Of course, when you start to learn to read, you do the phonological processing. However, it could be that when you become a fluent reader, that you sort of bypass the phonological processing and go directly to semantics. Again, there's a debate on this. But now with these tools here, we can also ask if there's parafoveal phonological processing, right? So you take in so we have ideas for doing that. That is, however, complicated by the fact that English and is there's a big overlap between orthography and phonology. That is not the case in Chinese. So we, in fact, now we are setting up to do a Chinese study where we can manipulate the phonology and it's not confounded by orthography. So there's more to come. So that was a long way around just saying that, yes, dyslexia is very interesting. Many people argue for a phonological component. So as a next step, we also want to look into the phonological component of parafoveal previewing. And that is also to gear up to do investigations in dyslexia, to have a full battery to look at both spatial attention, but also the phonological component and semantic processing. Yeah, thank you very much.